So, dear students, now we are going to discuss contributions of Julius Van Sox, and after that Cornelius Van Nile. First, Julius Van Sox. So, according to observations of Julius Van Sox, so plants are able to perform photosynthesis. They are able to perform photosynthesis in green areas. Green areas present in green structures was given. Nowadays, we are calling them as first one chlorophyll, second one is chloroplast. Green area where chlorophyll is present. According to Sox, it was not chlorophyll. Green color present in green structure. What is that one? Chloroplast. Okay, so during that experiment, he observed CO2 is converted into carbohydrate, that is glucose molecule. With the help of light, they are able to reduce a C of CO2 into glucose molecule. Means during photosynthesis, glucose is produced, carbohydrate, and that glucose, a number of glucose molecules change into starch. So according to Julius Van Sox, food is stored in the form of starch. Stored form of carbohydrate or food. Stored form of carbohydrate. So according to Julius Van Sox. Okay, so food is stored in the form of starch. Okay, so that's about Julius Van Sox. So at the end of all these, they prepared a empirical, unbalanced oxygenic photosynthesis. Empirical means unbalanced. CO2, not because of Sox only, so many scientists. CO2, H2O, chlorophyll, light. CH2O carbohydrate. Okay, so plus oxygen, O2 evolved. So that is a oxygenic photosynthesis. This is a empirical one, unbalanced one. What about the balanced oxygenic photosynthesis? So in the case of oxygenic photosynthesis, 6 CO2 molecules in the presence of 12 H2O molecules. Glucose plus 6 H2O plus 6 O2. This is a balanced oxygenic photosynthesis. Here the question is, what about the total number of H2O molecules utilized or used in this reaction? So CO2 is subjected to oxidation of H2O and reduction of C of CO2 into carbohydrate. So what is the role of H2O molecules? They are able to reduce C of CO2 into glucose molecule by undergoing oxidation. Okay, so what about the significance of H2O? Furtherly, we can understand that one at a time of light reaction. During light reaction, H2O molecules are subjected to photolysis, splitting of water. Go back to 10th class, one H2O molecule photolyzed in the presence of light. Okay, so half O2, okay, and 2H plus, that is oxidation of H2O molecule. So in oxygenic photosynthesis, hydrogen donor of hydrogen atoms to reduce C of CO2 is H2O molecule. So what about the fate of a H2O molecule undergoing oxidation during light reaction? We can understand that one at the time of mechanism of light reaction, not now. We'll see that one later. So that's about oxygenic photosynthesis. Now, against this, what about the contributions of Cornelius Van Nile, a microbiologist? He studied photosynthesis in green and purple sulfur bacteria. Green purple sulfur bacteria. Okay, so green, for here I'm giving some examples. Chlorobium, chromatium. With chlorophyll, they are able to perform photosynthesis. So according to Cornelius Van Nile, unlike oxygenic photosynthesis, in the case of oxygenic photosynthesis, who is the hydrogen donor? H2O molecule. Instead of H2O molecule, what about the experiment of Cornelius Van Nile in those bacteria? 2H2A instead of water. It took a molecule which is able to reduce CO2 into carbohydrate, CH2O carbohydrate, unbalanced. Empirical only, not balanced one. CH2O and 2A plus H2O. So this 2A is coming from H2A. So what about H2A oxidizable, undergoing oxidation? Here donor of hydrogen atoms is H2A, not the H2O. So that's why there was no evolution of oxygen. Instead of oxygen, 2A was evolved. That is a dummy one. Now what about the real one? Instead of H2A, take a H2S, hydrogen sulfide plus CO2, unbalanced reaction only, gives rise CH2O plus 2S, sulfur, sulfate plus H2O. So this one was the oxidation product of a hydrogen sulfide. So this is an oxygenic photosynthesis. Oxygenic photosynthesis. So what about the explanation given by Cornelius Van Nile? In certain number of prokaryotes, particularly bacteria, they are with oxygenic photosynthesis, anoxygenic photosynthesis. Reason, instead of H2O, here hydrogen donor is a hydrogen sulfide. So that's why there was no evolution of oxygen. So it is anoxygenic. Only oxidation product of H2S, sulfur was evolved, sulfur was produced. So there is an anoxygenic photosynthesis. Okay, and next conclusion, in the case of oxygenic photosynthesis, according to his observation, O2 was coming from H2O is responsible for evolution of O2. So in the case of oxygenic photosynthesis, O2 was evolved from H2O, not from CO2. So he demonstrated that in the case of oxygenic photosynthesis, the hydrogen donor is H2O. That's why O2 was evolved. That O2 is coming from H2O only, not from the CO2. So before Cornelius van Nile, so-called mechanism understanding of photosynthesis, most of the scientists believed that the oxygen is coming from CO2, not from the H2O. But according to Cornelius van Nile, O2 is coming from H2O only, not from the CO2. After that, he demonstrated, means he explained that O2 is coming from H2O only, not from the CO2. Later, not this scientist, Reuben and Kaman scientists, with the help of O18 radioactive isotope, O18 oxygen, H2O18, O18 radioactive isotope, they demonstrate that the O2 is coming from H2O molecule during oxygenic photosynthesis. So scientist names Reuben and Kaman demonstrate that the O2 is coming from H2O only, not from the CO2, with the help of O18 radioactive isotope. Just he proposed that O2 is coming from H2O, not from the CO2. He failed to demonstrate it. Who demonstrated that statement? Reuben and Kaman. So according to Reuben and Kaman, with the help of a radioactive isotope, O2 is coming from H2O only, not from the CO2. Okay, so what about the radioactive isotope used by them? O18. Okay, so that is about early experiments with the help of which we understood, and nowadays we are studying the mechanism of photosynthesis, right?